Hello everyone, good afternoon and welcome to this workshop on FAIR metadata. My name's Gary Saunders and I'm Data Director of EATRIS ERIC, and alongside me is Anna Niehaus from Radboud University Medical Centre, and we'll be the chairs of today's session. The focus of today's session is on metadata, and in this slide that hopefully some of you have already seen before, we're really looking at what powers the data that we see. So on this image of the iceberg, the data that we see is right at the very top, but it is powered by the metadata, and the structured metadata is so important to make the data that we see actually meaningful and usable. So this quite striking figure describes, I think, that the metadata really is what powers the data that we see. And on the next slide, Anna, please. The question is therefore, what is metadata? What is this whole bunch of data that we're talking about today? The first thing is that it's descriptive information. So information that can aid identification and discovery of the data that we see. We also have some structural information. It tells us how the data that we're seeing is organized, has been organized, and can be organized. It can also contain administrative data, so information about the technical source, how the data was produced, the data type that we're working with, and the creation process that one or a pipeline has gone through to generate the data that we see. Additionally, it can have some information about the reference of the information that we're seeing, so the content and the quality of the statistical data, the data provenance, this kind of thing. And then finally, it has information in the metadata. We have statistical information itself about the data, distribution, outliers, etc. And when we work in biomedical sciences and life sciences, next slide, please, Anna, the way that this is structured is around the FAIR principles. So the way that we can have metadata that is findable, accessible, interoperable, and reusable. So that the data that we see is indeed powered by metadata, but this metadata is structured in such a way that aids the usage of this metadata and ultimately the data that we have at our fingertips. So that's what our session is about today. It's about how can we better standardize and fairify, if you like, the metadata that powers the data that we see. So next slide, please, Anna. So in our program today, we have 4 sets of talks. The first 2 talks will focus on the multi-omics data verification process. The second 2 talks will talk about specific tools and services that facilitate the verification of multi-omics data. We'll then have 2 talks about the verification practices of omics data at data repositories. In our session, we'll then have a brief break. So we'll have a 10-minute break just before 7 o'clock. And then we'll come back to the final suite of talks to talk about interactive tools that capture and share metadata of multi-omics studies using templates. We'll aim to take questions after each pair of talks. So each speaker will have 12 minutes to give their presentation. And after each pair of talks, we'll stop and have a question and answer session. All people that are online should have access to both the live Q&A and discussion forum buttons. And Anna and I will do our best to keep a track of the discussion as it's happening there. And then at 8 o'clock, we will end the session. And what we'd like to get out of this session in the next weeks is that we'll write a summary of the things that we've discussed and what we've worked towards, and that hopefully we can provide a brief publication of what we discuss in this workshop for the community. So with that, I'll turn to our first talk, and that's Anna Niehaus from the Radboud Medical Centre. Anna.
All right. Thank you, Gary. I think my slides are no longer shared. There they are. So I'm going to start our workshop with the use case of multi-omics data verification from the researcher's perspective. This is one of 2 use cases. The next one will be presented by Veronica Suni. And I would like to start again with the FAIR principles that Gary just mentions and the corresponding guidelines that were introduced in 2016 to help create research data that is findable, accessible, interoperable, and reusable. And there are several guidelines about how to describe the data to increase its findability. These include, amongst others, the description of data with rich human and machine understandable metadata, the use of standardized and open metadata formats, the assignment of globally unique and persistent identifiers to both the data and the metadata, and the registration of this metadata in a public registry. Additionally, to make data interoperable, it is suggested to use metadata that is mapped to commonly used controlled vocabularies and ontologies. And following domain-specific standards is advised to increase the reusability by other researchers. So in order to apply these guidelines to multi-omics datasets, we have to consider various aspects ranging from the experimental study design to individual measured molecular entities. For understanding and reusing the data, it is important to capture the complete provenance of the samples and the data. Therefore, in the following, I'd like to give some insight into our process of creating FAIR multi-omics metadata. And since this work is part of two large collaborations, the Netherlands Cross-Omics Initiative and the IATRIS Plus project, I will first give a brief overview of these projects. So the aim of the Netherlands Cross-Omics Initiative is to establish a multi-omics research infrastructure across the Netherlands. And in the Cross-Omics data analysis, integration, and stewardship pillar, we work toward the realization of a so-called FAIR data cube that facilitates efficient multi-omics data integration. The Cross-Omics FAIR data cube comprises a set of tools and services bringing together different types of metadata, and associated FAIR data points serve as registries for the metadata and link to the different omics data resources. An important goal of this infrastructure is to enable also semantic querying of the metadata across omics data types and across studies. This work is being shown in different multi-omics demonstrator studies, which are realized by Cross-Omics in collaboration with different partners. And more information on this can be found on the Cross-Omics website. You can also follow the development of the FAIR data cube on GitHub. So today I'm mainly focusing on the European IATRIS Plus project, in which we are involved as well. And this project aims to support personalized medicine by development of IATRIS infrastructure and offering access to scientific tools. The IATRIS Plus consortium consists of 20 partners, including 14 IATRIS national nodes. And the main outcome of IATRIS Plus will be the multi-omic toolbox. And with the toolbox, IATRIS Plus aims to help overcome some bottlenecks in the personalized medicine and biomarker field, including resources to help improve technological and analytical harmonization, tools to facilitate data stewardship and adoption of the FAIR principles, and provision of reliable control reference values for biomarkers in healthy populations. The multi-omics toolbox will be developed and tested with a demonstrator based on an already established cohort of 1,000 healthy individuals in the Czech Republic. And this Czech multi-omics demonstrator cohort that we use in this project consists of 120 individuals as a subset of the 1,000 healthy individuals from the biobank. They range in age from 23 to 65 and have a balanced male to female ratio. And this data set comprises a number of different omics data types, which are acquired at different sites across Europe. These include whole genome sequencing, DNA methylation sequencing, mRNA sequencing, microRNA profiling, proteomics, lipidomics, and metabolomics. And in addition to the logistic challenges of acquiring the data, this also requires close collaboration for the FAIRification of the acquired data and the corresponding metadata. So the primary goal of our FAIRification efforts is to enable the integrative analysis across the omics types and also the preparation of this data set for future reuse. So when bringing together the various omics data types to describe this data set as a whole, we also need to bring together a variety of different domain-specific standards. And here we experience that it can be difficult for researchers to find the respective data and metadata standards from their field, and especially for omics types that they don't usually work with. And since it has become more common to combine different types of omics measurements in one study, this is a challenge that will be encountered more often, especially when we aim for machine actionable metadata. It is important to find suitable terms from controlled vocabularies, and this is a question very often raised by researchers. How do I find the terms that I can use to describe my data? And then once it is clear which data and metadata standards should be applied, often specific technical knowledge might be required in order to do so. In this demonstrator, we want to share our FAIRification process to get into a dialogue with the community. And with sharing this process and available resources as part of the IATRIS Plus multi-omic toolbox, we also aim to help other researchers to verify their own data. Additionally, we aim to share our computational workflows, the analysis workflows, in a fair manner, using principles. Sorry, do you still see my screen? Maybe I stop and share again. I have some problems with my screen. Gary, can you continue sharing the slides? Yeah, let me just check.
Thanks, Paula. That was my mistake. Anna, I'm showing the slides from my screen. We're on the verification of multi-omics cohort data and the goals. Just tell me when you want to go to the next slide.
All right, so I'm not sure exactly where it stopped. So I think it was mentioning the computational analysis workflows. So that is another goal of ours, to apply principles for fair research software that have recently been suggested to our analysis workflows and also share them accordingly. Can you go to the next slide? Thanks. So in order to describe multi-omics data, including all the different omics platforms, we chose the ISA metadata framework, and Felipe is going to tell us more about ISA later. But on this slide, you can see a part of the metadata describing an omics assay as part of an ISA tab investigation file, just as a short example of how it can look like. The reasons we chose ISA include the possibility to link different omics measurements to the same set of samples, the adoption of the framework by the community, and the support of readability of metadata for both humans and computers. And additionally, it allows us to link to published SOPs and protocols used in the sample analysis workflow and also in the data analysis workflow. Next slide. So with the IATRIS Plus multi-omics cohort data, we aim to integrate process data that is typically in a simple tabular format. And the ISA framework allows us to link each measurement to both raw and process data files. While the ISA framework itself does not support addition of metadata for individual measured entities, the ISA metadata can be combined with feature annotation files. An example for this is the metabolite annotation file or MAF format that is used by EBI's metabolites repository, about which Claire is going to talk later. And we thought about a possibility to add feature metadata to different and especially to processed omics data types. And the goals were that the format should be simple to use and allow automatic creation of persistent identifiers based on the given information. So our current version of such a feature metadata file is shown here. It includes each original feature name, a database identifier, and the corresponding database and its version. The assignment of unique and persistent identifiers to measured molecules make the data less ambiguous and also allow us to map identifiers across different databases, which is useful in the analysis as well as to understand the data. And so we share these ongoing developments on GitHub and appreciate any comments or suggestions on this. In general, we aim to use existing standards as much as possible. And then next slide. Okay. Yeah, and with this, I'd like to conclude with thanking our partners from the CrossOmics and the IATRIS Plus projects, as well as today's speakers and workshop participants, and I look forward to the next talks.
Thanks, Anna. That's a nice introduction to the IATRIS Plus project. There are a couple of questions. There's at least one question that's come into the Q&A forum, so I'd ask you to have a look at that and maybe write a text answer. And for now, I'll stop sharing my screen, which should allow Veronica to share her screen. Thank you, Paula. So let me introduce Veronica Suni. Veronica will talk to us about the ICANN Digital Precision Cancer Medicine Flagship Project. It's a real pleasure to have Veronica along with us and talk about this, this important project and the FAIRification of her metadata. Veronica, over to you.
Thank you. Hello, everyone. My name is Veronica Suni, and I will be talking indeed about ICANN Flagship Project. It is one of the largest projects funded by Academy of Finland, the biggest research funder, national, and it's run in collaboration with Helsinki University and Helsinki University Hospital. It's a large biobank project. So my presentation today is divided in two parts. First, I'll tell you about the project itself and the data platform, as well as the various data types that are collected and generated on the platform. And then in the second part, I'll talk more about the metadata and different types of metadata, so the biological samples and as well as sequencing data. That will be primarily transcriptomics and genomics. And in the end, a few words about the imaging metadata. Some of the most important facts about ICANN flagship project: it is one of 6 National Academy of Finland Research and Innovation flagships. It started 2 years ago, and already this year we are getting the first profiling data from the participants. It is a biobank project, and you see here Finland has quite a few biobanks, so we're lucky to collaborate with 2. One regional biobank, Helsinki Biobank, and another countrywide biobank, the FHRB, Finnish Hematology Registry and Clinical Biobank. ICANN is a public-private partnership kind of project, so it brings together researchers, more than 150 researchers working in Finland, and as well as working with industry collaborators, one of them being Boehringer Ingelheim. The aim of ICANN flagship is to provide solutions to precision cancer medicine and build an ecosystem by 2026. And we believe this can be achieved by integration of omics and other molecular profiling data that's generated in the project with extensive health registry data from the same patients. And that's done on a large scale, so I'm talking about thousands of cases. We're estimating that by the end of next year, we'll have samples from more than 5,000 cancer cases and more than 12,000 cases in total. And ICANN is truly a pan-cancer project, so here I'm listing the areas and the cancer types we'll be working with. So all this data will be processed, integrated, and analyzed on an ICANN data platform that we're working to develop. For all the patients that have signed the consent and are part of the project, there will be extensive clinical data collected about them. We'll include the diagnostic data, patient visits, patient imaging data, and all of that will be taken to a secure environment, a data lake hosted in Microsoft Azure cloud services. In addition to clinical data, there will be samples taken and various profiling will be done on those samples, and I'll tell you in a moment more about that. Finally, once the molecular profiling data is acquired, it's brought together with clinical data to ICANN Discovery Platform. That is a GDPR compliant environment, and it also aims to be fair. So we're offering all these different types of data, the research data, registry data, imaging data, and offering it for use for different levels of users. So we are prepared to cater to data scientists and researchers and offer tools accordingly. And one of the things that makes ICANN data platform and data meta fair is the fact that we are using the common data representation. So the clinical data is harmonized and structured in a certain way. So we're following the OMOP data model. For those of you who are not familiar, OMOP stands for Observational Medical Outcomes Partnership, and really offers us a unique opportunity to use the numerous tools developed specifically for that. There is a module, oncology module, with numerous attributes specific to cancer that are well defined. With person or patient, they're always in the center. An important bit here as well that it's all in English. We're dealing here with Finnish health system, with some records, of course, being in Finnish, but once we start using the complementing OMOP structured data, we're no longer limited to the Finnish text mining and tools and professionals and researchers working with that, collaborators as well. So the molecular profiling that's done on all the samples that come in, those thousands of patient samples, can be split into two. So we have a general molecular profiling on top and then the deep molecular profiling. So the general molecular profiling, almost like a package, offers for every sample, for all the samples in the pipeline, whole exome sequencing, RNA sequencing, and basic immuno-oncology is done that. So multiplex immunohistochemistry imaging and immunoncology analysis from sequencing data. So that's collected for every sample. But then there would be a subset of samples that would be analyzed further. And that's done according to sub-project needs. There are more than 20 sub-projects in ICANN at the moment, and the project will still continue quite a few years. So that's why here I'm listing just some of those analyses that are planned, and I'm sure that this is totally incomplete list, so some more will appear. So we have here single-cell RNA sequencing, ATAC sequencing, proteomics, metabolomics, epigenomics, ex vivo drug screening, and others. So finally, we arrived at the metadata section here, and just wanted to say that we also planned and wanted to follow the FAIR principles from the start already before we had the data. And I think we succeeded the most in the reusable part and the interoperable part. So we do have extremely richly described metadata, focusing on collecting as much as possible, or rather more than less. And then we try to meet the domain relevant community standards. So as much as possible, reuse the existing vocabularies and ontologies, and certainly made sure that vocabularies that are available would follow FAIR principle and would take that into account. So this was a long introduction to the first talking about the biological sample. So lucky for us, we're working with biobanks that keep great track of sample origin. So that is available in detail. And there were as well existing vocabularies, ICG, CRGO, and SNOMED-CT, so for the tissue source of best specimen and tumor normal tissue origin, as well as anatomical source. We used those vocabularies. And here I'm mentioning a couple of things that could be characterized as challenges. So mapping what has been recorded and is available in biobanks to FAIR model, and as well working now with 2 biobanks, and the project will grow and other biobanks will join. So we are forced to work with slightly different systems. So we have source system differences, and that's okay. That wasn't such a big of a challenge. Now, metadata for the sequencing data. We wanted to include, of course, the study design, sample preparation, sequencing, as well as the analysis. And here it's our schema is kind of a mix of EGA metadata and FAIR Genomes project, and we're adding and removing some terms based on the feedback received from service providers and researchers. And as a basis for vocabularies, we used EFO ontology. And so we were trying to have our metadata schema tying the sequencing metadata together with the analysis results, and have it in an easy to query way. So here's a snapshot of a sequencing part of the schema. The goal has been to make it modular, so it can easily be extended when the new analysis types would appear. Here, a bit of an extension, spatial transcriptomics has been added, ex vivo drug screening, tissue sample information, and single cell preparation, and a few others. So in the end, about the imaging metadata, remember we had this general and deep profiling. So part of the general profiling was multiplex immunohistochemistry imaging that exists on several levels. So the metadata we need to collect would be multiplex staining, imaging, sample information with multiple samples on each side, as well as the analysis. And until very recently, my colleague corrected me. There actually are some papers from this year. There were no existing standards for multiplex immunohistochemistry data. So we were inspired by the open microscopy environment standard. And this, I guess, is a challenge that we are trying to solve right now. So creating a schema that would correctly label all the hundreds of files on all the axes, and it would be both machine and human readable. In summary, I told you about the ICAN project. It's a large Finnish biobank project, pan-cancer, and it's a discovery platform. And what makes this project special is that it will allow the integration of omics and other molecular profiling data with very extensive health registry data. And with metadata, while we're nearly done with metadata specification for the general profiling, there's still a lot of work that remains. So the question will be, the existing standards and vocabularies are there for remaining data types for the deep profiling part of the project. And we would like to create a schema that ties together all the 24 or more modalities, data types, the sample information, and the analysis results. And we'd like to have in the end an external resource as well, where metadata can be queried. And I finish with this slide listing all the people who have helped us develop the metadata specifications, especially my colleagues from ICAN core data team, Anna Kuosmanen, Tamara Lack, and Esa Pitkänen. And I'd like to thank you for your attention. Happy to take questions. Looking forward for the remainder of the workshop.
Very good. Thank you, Veronica, for the presentation about the ICAN platform. So the main way that we'll take questions during the workshop is via the Q&A forum, which you should find at the bottom tab of this ECCB platform. So please, if anyone would like to type any questions in there, then we can ask them directly to the speakers in the few minutes that we have. First, Anna, can I ask you a question related to the IATRIS Plus project? So the metadata schemas that you've finally settled on are quite complex. And I'd like to ask you a process question. You know, and within the consortium, what was the process by which you decided on which metadata schemas to settle on? And how did that conversation happen? Was it IATRIS Plus specific, or did you take inspiration from other projects? Did you take inspiration from other things that are happening in other places? And how did you settle on the final list of metadata standards that you wanted to try and use?
Yeah, so first of all, for the individual omics technologies, we apply the existing community standard formats. But this work also followed up on what we have already done within the CrossOmics consortium, which also builds upon the FAIR Genomes project that was mentioned by Veronica, to have a starting set of vocabularies that is relevant to our data, but also similar to what Veronica mentioned, this is still an ongoing process to find what fits best to our data and what is already used so that it is interoperable with existing data.
Thanks, Anna. And then Veronica, leading on from this question a little, in your presentation, you talked about the mapping of metadata schemas to legacy data and the application of metadata schemas to prospective data. Can you give a little indication to the major challenges that you see when you try and map legacy data to metadata schemas, and the comparisons between mapping to legacy data and preparing metadata schemas for prospective data? And, you know, for thinking about the crowd here and some of the questions that we've seen, where the main challenges lie in both cases.
Yes, so I must say right away, I wasn't there 2 years ago when the project started. So the metadata specification that I presented today and many of the decisions that were made without me there. But I'm now participating in specification of these new data sets and new data types. And we see that it really takes numerous iterations. So we meet and then we meet again, and then something comes up as something totally incompatible. We cannot have it like in a standard way for different teams to have it. So we want to have as much as possible. We cannot really collect everything we'd like to collect. And there are also technical challenges, I'd say. There would be systems that are quite close to the information is available, we'd like to have it, but getting access to it is tedious, complicated, and sometimes one wonders, is it even worth it? But it may be quite needed. So I guess that would be the biggest challenge, in my opinion, is data extraction, kind of hard to reach out. And then because we're dealing with patient data and it's very sensitive. So there needs to be a compromise.
Yeah, thanks. And then one final question to you, Veronica. So you mentioned the OMOP framework within your presentation, and you also referenced 20 projects within the ICANN consortium. So can you comment on the uptake of the OMOP specification and framework within these projects, and how you see that for the ICANN consortium, or maybe even wider within the Finnish suite of projects itself as a country even? Would you be able to comment on the challenges and the opportunities that have happened through the application and the uptake of the OMOP framework?
Yeah, so luckily it is not my challenge. I know this will happen. There is a whole dedicated team working on this, and I know it's a lot of work. Yes, the promise is that we will have OMOP data for every patient. So absolutely, opportunities are excellent, because the limitations frequently come from this language-specific and very different systems as well. And here you get one allowing you to mine, on a national level, so much information. And like I said, this is now 2 biobanks, but the plans are to extend. So this was one part of Finland that was mainly covered, but it will be wider. Yeah, great. It offers great opportunities. Yes.
Yeah. So from the European landscape, where I work, there's a huge focus right now on OMOP, and I really like to hear the opportunities that you see from Finland, because it's been replicated in many different countries, many different projects right now. Okay, thanks a lot both for your presentations. I think with the time, we'll now move on to the next suite of presentations, which will talk to us about tools, services, and resources that are available to facilitate multi-omics data FAIRification. And the first presentation we'll have in this suite of presentations comes from Alison Lister of FAIRsharing. Alison, I can see you on the screen, and hopefully you should be able to share your screen and operate the slides yourself. If you have any problems, just let me know and I can hopefully share from my side.
Thanks, Gary. It looks like the slides are up, so do let me know if they ever disappear. Thanks. Perfect. So thank you for having me. I am the FAIRsharing Content and Community Coordinator, and I'm going to talk to you today about how you can use FAIRsharing to discover and curate a whole ecosystem of research standards and databases within the multi-omics research domain. So first of all, just a very, very brief glimpse into what FAIRsharing is for those who might need a refresher. Well, we provide curated descriptions and relationship graphs of the standards, the databases, and the policies that are present across all research areas. So that means the databases plus the standards they implement, data policies, and the databases they recommend. It's all linked in a network of resources and relationships. So it looks, oh yes, it finally all drew. That's great. So we have over 3,600 resources. Apologies, I just realized I used a slightly older version of my slide. And we have 2 main ways in which people enter FAIRsharing. First of all, as a consumer of the resources at FAIRsharing, that means that they're coming to look for a database or a standard or a policy to help enable FAIR data. Or they could come to us as a producer, that is a database developer or a standards specification writer who wants to describe their resource with us in FAIRsharing to make it more visible, more widely adopted, and more cited. So we have a lot of different communities and working groups that we work with to try to make sure fair sharing is adapting and listening to the community that we serve. And we're a member of a number of different working groups within Force 11 and the RDA, and also a recommended interoperability resource with Elixir. So moving past this onto a use case that might be a little bit closer to your heart, this is also present in our documentation and as well as within the slides, so you can look at it later. Apologies, but I'm going to use the Adventures of Alice and Bob. And I did try to find a stock photo of Alice and Bob and see if I could find some good representation of a scientist, but I just got increasingly enraptured with these lovely stock photos that people are having about that are supposed to describe scientists to you. So in the end, I found something that was reasonably close to who we're trying to describe. Okay, so we're describing Bob, who is a researcher, and Alice, who we're imagining is a data steward or a librarian. So Bob has come to Alice because, let's say, his funder, maybe the ERC, has requested that all his data for his upcoming project should be fair, and he's not quite sure how to get started. So he has a few meetings with Alice, and Alice shows Bob various ways he can find the most appropriate standards and databases for his data type. She describes, for instance, how fair sharing can help him identify databases for depositing his data or finding existing data relevant to his work. He also learns about identifying standards through fair sharing for describing and reporting his data so that it's more understandable, reproducible, and reusable. And finally, Alice shows him how he can discover data policies from journal publishers, funders, and other sources that recommend specific databases and standards. So we have Bob, who tells Alice that he wants to explore his funder's data policy in more depth and is also looking to discover any resources within multi-omics or just any kind of omics domain that might not be explicitly listed by the funder. So Alice explains that the first step here would be for a researcher who has a policy that they should conform to. They need to see if that policy is in fair sharing. So we can use the search tool to find the ERC policy. And this policy, of which this is a screenshot of the fair sharing record for the ERC data policy, allows them to review general information about it, contact the maintainer of this record, which is the ERC itself. All of our maintainers in fair sharing are the developers of the resources themselves. And also they can click through and access the policy itself if they need to refer to it. And this is just the very beginning of a fair sharing record. It shows some general information and some subject tags, as well as the DOI for the record itself and a description. So if we move on, what it also shows is further down the record is how to cite it, of course, but also the relationships that this data policy has with those standards and databases that it recommends. In this case, depending on how good this screenshot is and how good the browsers are that you're viewing this through, the ERC data policy has 24 explicitly listed recommended databases and 1 related or recommended standard. And it makes a graph to show you not just those resources recommended by this policy, but also how those resources interrelate. So really, relationships for me are at the core of fair sharing. And if that's one thing you take away from the Adventures of Alice and Bob, it's that the relationships are what's most important for us. So what you have is a way to describe the whole ecosystem, not just isolated resources, and how they work together to build a community of resources that matches the community of researchers that are building it and contributing to it. So Alice and Bob can now explore the databases listed explicitly in this policy and use the metadata available in each of the records to determine the best place for Bob's data. However, remember that fair sharing can only reproduce explicitly named recommendations. So policies often have a catch-all section, in this case the ERC does as well, stating that any fair enabling resource can be used. And Alice points this out to Bob. So are there other resources out there that Bob should be aware of? Well, what we can do is we can search fair sharing. And what they do first is they're searching based on the subject area. So there is a lovely graphical display of all of our subject hierarchies, of which this is a screenshot. Again, apologies for the clarity. You can either drill down according to the name in a hierarchy of text, or you can use our sunburst diagram. So here they're drilling down to the omics part of the fair sharing subject hierarchy. And from this summary, Alice and Bob can click through after they've looked and seen that there's 781. Wow, that seems like a lot. Now that's 781 mentions of the word omics or any of its child terms, because we use an ontology to describe our subjects. So you can click through and access the main search results. And you can find that ultimately, because there was some overlap in the tagging, there are 525 records tagged with omics. So result sets can then be further filtered according to a number of different features that you might want. We have a wide variety of resources. Alice and Bob are pretty happy, but are there any further requirements that they could use to further refine? Let's just see how the standards implemented by these databases can help inform Bob's decision. So as an expert, Bob is aware there are a number of community standards that have been developed over the years, and so he refines his search. He chooses only those resources that have a ready status. He also chooses ones that have a particular, we might look also for this specific omics tag within a description to see if that gives us a little bit more of a hit. And also you might want to be looking at a particular type of standards. He notices in the search results, there's the omics DI XML format. Whoa, he says, this seems like a great thing. And indeed, the description of the omics DI format looks promising in fair sharing. So he takes a look at the records graph to see those resources, that ecosystem surrounding it. And there's a number of both specialist and generalist omics databases that implement this format, including the omics DI resource itself. So Bob now has a lot of different choices and can take a look and find the resource that best suits his needs. To finish off, I want to talk a little bit about what fair sharing can do for you as a community, as opposed to an individual group or researcher with a set of data. So what we have and what we offer to the community are tailored views for education and promotion of your community. And these are called collections. They're branded pages that group standards and repositories together according to a particular criteria that you decide. And initiatives and projects have created them for several purposes. For example, to list resources developed by the community or developed by a particular standards organization. To list resources recommended by a community rather than perhaps developed by them, and even mapped to each other. So a live version of crosswalk resources listed in a publication, for example. And I told you a little bit about the knowledge graphs and the relationship graphs that fair sharing has for every one of its records. This shows you a slightly more hairball version of one of these relationship graphs, because I wanted to give you an idea of how a community has implemented a collection in fair sharing specifically for this purpose. So the ISO biotechnology standard has 256 different standards listed. And of course, it being ISO, once it's all ready, the full version will be behind a paywall. And so how can you have a look at it and also see what happens, because ISOs update every few years. Fair sharing provides a live version of this ISO standard developed together with the authors of the specification that shows you which ones are ready, which ones are in development, and which ones unfortunately are deprecated. We also provide machine accessible content. So when you describe resources with us, you get the use of our API and a number of different fair evaluation and assessment tools, as well as DMP management, things like this, use fair sharing programmatically to help inform their resources. So when you add with us, you gain access to that whole tool ecosystem as well. So we as said before, and in summary, we provide a lookup service for fair evaluation, DMP, and other tools and services. We are a searchable registry for people, and we allow the creation of community profiles, these collections, so that you can really shine a light on the work of your organizations. And I just want to finish by saying thanks very much to the whole team and to our executive and stakeholder advisory boards and to our early adopter community curators, which I didn't have time to talk about, but which if you're at all interested in what we are developing and what we've just started up as the Fair Sharing Community Curation Program, I would be happy to talk about it with you later on. Thanks very much.
Very good. Thanks, Alison. That was an excellent whistle-stop tour of a very complicated resource that serves as a registry for a number of different services, policies and standards. And I myself have a few questions, but I'm going to hold off until we hear from the next presentation. And if we turn to the next presentation, that'll be from Philippe Roccosera, and he'll talk to us about his experience over the last 10 years with ISA. So hopefully, Philippe, you can share your screen and manage your slides.
Yes, I hope you can see my screen. And hear me well. Thank you, Gary, and thank you, Anna, for organizing the event. Really a pleasure to speak today. So yes, it's really nice to be able to speak about ISA, and I'll try to give you a bit of the long view, and maybe going back to some of the questions we had about the FAIRification and how ISA relates to FAIRification. I think ISA will focus specifically on making sure that community requirements and metadata about experiments are reported. So this is one of the requirements of the FAIR principle, which is related to communities mostly. But before that, I'd like to give a bit of a insight about where ISA came from. So I'm old enough to be able to do this kind of exercise. And basically back in 2002 when we joined the EBI, we were already confronted with studies which were multimodal. And that, in a way, presented a number of challenges at that time for several reasons. The first one was that a lot of the models were technology-centric. So we had, and ISA stands on the shoulder of giants, really. There was a MAGE object model, the microarray gene expression model, and that was really solely DNA microarrays to report the starting of the transcriptomic profiling adventures. We also had the PRIDE XML and the MZML for proteomic and mass spectrometry. And that was nascent. There was the SRA XML, so the short read archive for the INSDC databases. And as you can see, this is already presenting a potential problem because of the subtle difference between each of these models, even though some of the elements were covered and common to all, like the study. You always have to describe a study or a protocol and these kind of things. And also what is interesting is that some of the models were actually implemented only by one repository and not the others. For instance, if we compare the US and the EU repositories, the MAGE object model was not implemented by GEO, for instance, in the US. It was only ArrayExpress and the Stanford database, for instance, at the beginning. For ENA, for sequencing applications, it's a bit clearer because INSDC has done a great job, and SRA and ENA are using the same schema for the data, but that was like that. For proteomics, it was a bit more complicated, and for metabolomics, there was nothing when we started, and so this is where, in a way, we have this kind of privileged relation with the European repository for metabolomic, and Claire will tell you more about this. But also, in a way, the situation was not that bad because they were existing efforts. I mean, this is interesting. I said we were working with a lot of groups that are actually working on multimodal studies. At that time, it was mostly toxicogenomics, and we had collaboration with NIHS in the US, so the SEBS database, and they were looking, as you can see from the publication, to integrating data coming from transcriptomics and proteomics already. And of course, there was a development of a new model, which is called the functional genomics object model. And interestingly, I worked with Alison, who talked just before me, on creating XSLT transformation to go from the XML message that was produced from this model into tabular representation. And that leads me to the next point, which was, again, why we created ISA, or ISA came about. In those days, the format that were mostly manipulated were fairly large XML documents. Large, I mean large in 2003, which was like 100 megabytes, for instance, which was difficult to parse. But that was, again, the difficulties on technology and maturity as well. And basically, for wet lab people, I mean, it was extremely hard to open an XML file. So that was the reason why we need a presentation layer. And this is where, using the technology stack, we looked at XML. Okay, we can use XSLT, which is a transformation language, which allows you to convert an XML document into PDF or into table, HTML or text table. And in a way, as we did this, people picked up the tab format because it was, in a way, easy to produce, seemingly easy to produce. But then this is where you run into complication. You run into complication about enforcing requirements, enforcing annotation requirements, and validity of the table that are produced. So then really you're caught back by the hard realities of computer science, and validating the message is critical to infrastructure. And so these are the aspects of the kind of crosstalk between the users and the practitioner, the developers. And also there are evolution in trends. There was a rise of Python at that time and JSON formats. So when we looked at this, we took this into account. So we generated a set of tools to support the ISA specification. The ISA specification is fairly simple. It's a graph that connects the different type of nodes. We have material nodes, so you can track the workflow that transform a material into another material, let's say a blood sample into an extract that you are ready to run into an assay, for instance. But you can also track to some extent some of the data transformation step. Typically, the kind of, if in the case of a multi-omic or of a transcriptomic assay, you will have to run to acquire the raw data and then normalize that. So the step of normalization can be tracked. You've got a pooling effect. And the reason why I'm mentioning this is because we developed a series of tools. The initial tools were developed in Java, and they were the ISA creator software tool that you can see here. I'm showing here an interesting plugin, which was a glyph-based representation of the underlying graph. What is interesting with this plugin that Eamon Maguire developed in a group when he was there was the ability for two things. First, it uses a new novel paradigm for representation, the notion of glyph. The glyph gives you a story about, so the diagram that is created depends on the annotation that you find in the file. So that was a way to try to incentivize people to give us more metadata by allowing, if you give us more, we are able to produce a nice figure for your publication. So we were testing in those days the kind of options, how to incentivize and reward people spending a bit more time in providing more metadata. So that the data, as Gary mentioned in his introduction, can really be understood by third party. And this is the aspect of reuse in FAIR and also to some extent interoperability. More recently, we've been focusing on developing an API because we realized two things. People didn't like necessarily to have a desktop application, and they would like to develop their own layers of either database or web application, but they wanted to have support to parse, to manipulate the data structure that we were producing. So this is the ISA API. You have the reference here into the GigaScience article. And the key salient features are the fact that the ISA API allows you to create an ISA document. It can be completely granular, or you can use the information about the study design to prime and kickstart the creation of a full ISA document. And then we have the support to convert and validate. Well, validate first and then convert to format that are accepted by the public repositories like Short Read Archive, Mage. And this is important because, again, it's not creating another incentive to shield people from some of the aspects of the submission, while also providing the ability to support them when they are faced with managing multimodal studies. So moving on, we now have we support two different sterilization. One is the tab, because in a way still the ability to have a tab delimited random format makes it easy for people data scientists to run standard tools like pandas or tidyr. You can manipulate these data frames fairly easy. The trick is that the tab, the presentation, the information, the ISA information in tab is also actually a graph, so it's not completely neutral, and you have to bear that in mind. And this is why we had to have the JSON representation, which actually represent fully the exponential graph, the one that we were showing the picture a bit before in the ISA creator tool. Also, the advantage of having that in JSON is that you can validate against a JSON schema, which gives you, again, ways to detect departure from optimal. And this is also an incredibly important aspect of interoperability when we are building services and systems. And this is where, again, we have to go back to validation against hard schemas, and which is equivalent to what we had in XML in a way. And of course, we use Python because this is a language that is for the API extremely popular, and JSON because it's a data structure that is extremely popular for web development. Again, trying to keep in mind that people would like to roll their own user interface and presentation layer, so we don't want to prescribe anything. So now I will switch gear a little bit and tell you about the adoption and the community. I think this is what has been really nice, is that in a way, ISA was addressing a need. It was, I mean, from the talk from Veronica and Anna, it was obvious that there is a need for a format that supports multimodal studies. I'm not saying ISA is perfect, but it's one solution that is available here. And here you've got a number of collaborators from EMBL, EBI, Metabolites for metabolomics, to FAIRDOM-seq, to the Jackson Lab. More recently, we have the NATA-Gene Lab that's using ISA and some of the tools as well. And you can see, obviously, CrossOmics with Anna. What is interesting also is that we have a diverse range of community. Plant scientists as well have been using ISA. Like we've got a tool collaborators at the Ehrman Institute with the COPO project, which developed a complete system for managing sequencing data. And more recently, we've been involved in a grant with Gosmic Lem in Cambridge on the ISA Intermine. So of course, it's a very live community. I included the links to the ISA API GitHub repository, where you can track issues or initiate discussions. We have also a Google group discussion. So please, if you have any issue, just get in touch with us. Also, another sign that the community is quite active is that if they don't like your software, they will write their own software as well to do this. And we have two examples here. We've got ISA4J at IPK. This is again plant scientist, and we have it's featured in our blog post. And this is really because they had a need for having very fast read-write operation, because they deal with thousands of read-in samples. And this was not, the performance of the ISA API was not there at that time for them. There is another implementation, similar idea, but again, this is also, it's optimized. They can go quick because they're optimized. They only support a subset of the ISA specification and only the tab. So that simplifies a lot the workflow. And so that's, if you have this case, I wanted to point out these resources. In the last few minutes, I think I will go, more on the aspect of the community which is about creating configurations for ISA. ISA is very generic. It doesn't specify the organism or anything like that. It gives you a framework for tracking the workflow in the lab to some extent. But as soon as you have a species-specific or a thematic theme-specific, you need to be aware of, okay, maybe implement configurations so that you can ensure that minimum information checklists are implemented and can be used by your tools. And we can see that already with even when we need to extend the ISA to support a new type of assay. As Veronica pointed out, it can be demanding exercise to understand the technology and then fold it into a model, identify the vocabulary, the value range for the value sets for a particular value, and all these kind of things. So we had kickstarted this effort. We had a number of configurations, but it's extremely hard to keep up to date all the time, also because we are a small team, I guess. And it's also a large community, so we cannot be everywhere all the time. So I think this is what we would like to convey, is that we should try as a community also to, in a way, see the patterns and implement things together, and notify each other when there are things that are not, maybe that need improvement or updates. And this is interesting. We will have a meeting at DBI with the NA team to review and revisit all the ISA configurations supporting sequencing applications, so we can be sure that the SRA converter is operational and up to date. Then, of course, documentation is key. It's extremely important. We have a list of Jupyter notebooks that show how to use the API and the key functions. I don't have much time to go into this, but I included the link. Then I will finish on this aspect of on FAIRification. In a way, we started. We have the ISA tab, which is easy to use. We can be free text, but then you can make it more fancy. You can start annotating the free text with ontology terms, and still within ISA tab. And then you can convert this ISA tab into ISA JSON, which is more machine readable and palatable for agents, software agents. But you can go even further now. We have developed a series of JSON LD context files, which are actually for either the OBO Foundry, but also schema.org or Wikidata, which means that you can choose which conversion to RDF you may wish. And if you, for instance, choose to have schema.org, that already gives you a fantastic opportunity to improve your discoverability through search engines if you post that into your web page, for instance. So this is what I don't have much time to delve into this. We are also looked at with collaborators to see how we can package the information of the metadata coded into ISA, pointing to the raw data files, using frictionless data matrices to show the associated results, the actual findings, and package everything as a research object, and possibly give it a Globus Mini ID, for instance. That would be one aspect on how to do FAIR. I point to, in this slide, to a resource in the FAIR cookbook, which details how we can do this if you are interested. And I will finish with a recent development on the ISA API. I think we've did over the summer with Dominic Battista and our team a huge amount of work for refactoring the ISA Python package. We improved the performance drastically, improving the test coverage, adding data sets. We have the study design create mode, which again is interesting. It supports factorial design and also repeated measure design, so more longitudinal kind of designs. We have GraphQL interface now that will allow to create the ISA documents. And as I pointed out, we have this RDF linked data coming along with schema.org, Wikidata, OBO Foundry. We also have a couple of experimental converters now with metabolomic workbench and Biocrates. So if you're doing metabolomics and targeted metabolomics, that could be of interest. So yes, to finish, Tecom, this is, I think the key point is creating this community of practice and around ISA configurations. And this is coordination. This is hard, but I think this is necessary to ensure interoperability for the community that uses the ISA and the convergence. So hopefully we'll find to organize meetings, depending on funding opportunities, but that would be the project for the next year. So I'll finish with thanking the organizers, so Hannah, Gary, and Manuela. Really thank you for the opportunity, and the people in my team, particularly Dominic Battista and Susanna, and also the ISA community. And this is fantastic. Thank you all the people that I've. So I'll take any question now. Thank you.
Excellent. Thank you, Philippe, for this overview of the ISA format and its current status. I really like the emphasis on the validation. I think when we think about metadata schemas, the validation aspect is really key. If we're looking at questions, Alison, when you were presenting, something really struck me, and it's something that I'm seeing in multiple projects and multiple consortia right now, and that is the want to try and present a user journey based on standards and policies and how these can connect. So from the graphs that you presented and the ways that you can find things based on the policy to connect different services, is there a way in fair sharing to show a user journey based on a specific use case, and say, if you started at this end of the pipeline and wanted to get here, here's the services and policies and standards and different things that you could use to connect services and actually get to an output. Is there a way to do such a thing?
At the moment, the only place it is the documentation that represents this presentation that's in our Git book. But you ask a very timely question, because some of the work that we've got planned for the next 6 to 18 months is all around the user journey and exploring the sort of visual storytelling and the sort of dynamic movement of different user roles through fair sharing in lots of exciting ways. So we have a new developer that started a few months ago, and his primary area of work will be around this. So do keep us in mind over the next few months, and I'm sure we'll be asking the larger community through Twitter to provide their thoughts on early versions and things like this.
Perfect. Thanks, Alison. I also note, Alison, there's a question in the question section for you, but I'll leave you to answer that one via text if you can, please. So we'll move on and keep with the schedule. So thanks to Philippe and Alison. We'll now move on to the third pair of talks for this workshop, and that's on the FAIRification practices at omics data repositories. And we have representation from 2 EBI repositories here. We have, first of all, metabolites via Claire O'Donovan, and then we'll talk about the PRIDE database from Deepti Kundu. So Claire, if I can ask you to share your screen and then navigate your slides. That would be nice to hear updates from the metabolites database. Thank you.
Excellent. So again, thank you for the organizers for giving me this opportunity to talk about our resource. If you haven't heard about metabolites, it is the home for metabolomics experiments and derived information. And this is just another variation on a slide you've all seen before, the FAIR guiding principles. And I have been attending meetings since early 2017 all about this. I guess my key message, which echoes what we've heard from Finland and from Philippe, is that things keep changing. So one thing to realize is that metabolomics is one of the newest omics. So when until 2017, the metabolites database was very much a research effort. It was to put gold standard studies in there for tool development and to help build studies. But it wasn't really a repository, as there wasn't that much of a tradition of data sharing in the community. That's completely changed now. And we wanted to learn both from our colleagues at the EBI who've been in all these omics things for years, as Philippe presented as a EBI alumni from some time ago. We wanted to learn from other people. But the metabolomics community itself, there is a society and it was very, very active. And so there was the Metabolomics Standard Initiative. And this was taken very seriously in 2012. Lots of papers were published. They were used to provide those gold standard data sets in the early version of metabolites, which was released in 2012. And then, I hate to say it, but they would agree with me, the community pretty much forgot about it. It was one of those efforts where we all agreed that well, I wasn't involved back then, but the community firmly believed, as Philippe said, in agreeing standards and the kind of data that you needed to capture. But when it came to actually submitting it to open resources, there wasn't that much interest. And the other thing, which is quite different to the other omics, is that metabolomics comes from the chemistry family, really, and they didn't have the same attitude towards data sharing because there was so much industry involved. There was a very different mindset. I've spent many years working in genomics and proteomics, and there was always much more of an openness to sharing the data after people got over their initial things about trying to patent human genes. Once that was over, people had very much more the attitude that data should be shared. So Metabolites now is an open source study repository, and we're building up the metabolite knowledge base. That's the other thing that you really need to know about metabolomics, is that, and I know that genomics people don't like me saying it, but it's much, much harder to figure out what data you actually have, or predictions. And I've been working in bioinformatics since 1993, so I do remember when this wasn't trivial. Actually knowing what you have in your samples is really hard. And one of the reasons why that's hard is because references to know whether your compounds are not freely available. People actually have to pay quite a lot of money for them. And so often experiments are done where compounds are present, but they're not looked for. And so that information is missed. So I put in this slide to stress to you that our community is really, really diverse. So you have people who are doing metabolomics in labs. You're doing it as part of disease control. You're doing it as part of the NHS. In the United Kingdom, we've seen how it's being done in Finland. There's also companies. Metabolon is listed here, but Philippe also mentioned Biocrates. All of these. And then you have the epidemiologists who've totally gone into their business in the last few years. And so all of these people have to submit data, but they also have to deal with the restrictions that apply in each field. And of course, human data has many restrictions, as we've all learned. So I popped this in to show you that while in theory, we are the European and we are the Elixir recommended repository, our data is actually global. And China is our biggest submitter, followed by the USA, which is, again, putting diversity into our user base, because we can't just use Elixir to understand what our community wants. We have to deal with an international community. The Metabolomics Society is helpful for that, but we do have to keep many hats on. The other thing you'll see there in the submission rates, so in 2012, we had 12 submissions, and last year it was over 1,000. And the team has not grown in that time. The other big change that has happened is that originally there was quite a lot of plant and model organism databases present, but not so much human and mouse. Now the database is half human and mouse studies, which again means that we've had to adjust and learn. So one of the reasons, oh, and then the final little box there is to show that metabolomics has very much transitioned out of the chemistry area that it was originally in, and it's now OR-specific metabolomics, but is far more represented in general publications such as Nature and PLOS, which again indicates how widespread metabolomics is now being done, if not always in the best way. So again, the database is cross-species, cross-technique, and you've had a little bit of detail about how complicated that is, technology-driven. A lot of that has to be taken into account. And the other key thing is we want everything to be open access. So while we do have colleagues in the States doing Metabolomic Workbench in San Diego, and Metabobank in Japan, which just started up in the last year or so, they weren't so insistent on the raw data. We insist on all of it. But for the purpose of this talk, what I would really like to highlight is the importance of ontologies. So one of the first things we did when we redeveloped the metabolites database in 2017 was introduce ontologies. Some of you on this call, probably mostly the speakers, will be absolutely shocked to understand that there was very few ontologies actually used, not even the NCBI taxonomy. So a huge piece of work was done by lots of people, and I give them sheer credit getting their heads around something completely different and implementing a really nice approach. But the key thing here is that this continues to evolve. We want to continue to work with communities, including obviously the ISA. And Philippe, we have a call with him on Wednesday to iterate again. And I guess that's my overwhelming message, is that you're never done. So all of this ontology mapping that we did and we have built on over the last 5 years, we're now starting to completely re-engineer again, because we have so many studies. There are so many things we want to capture in an ontology way that it is really, I think the UK expression is painting the Fort Bridge. It never stops. But that's all the more important, that we all work together so that we're not reinventing the wheel, that we all learn. For instance, MSIO there on my left-hand side for instruments, that's Philippe's, and EFO has been mentioned. We all need to contribute to each other. So we do contribute to EFO when we find new terms in our community. We're not in the business of creating our own. So there's a lot to cover in metabolomics in 12 minutes. I just wanted to highlight the kind of data that we capture, and I see a spelling error there already. Descriptors, protocols, samples, assays, metabolites, all of that information we try to capture in a consistent way. And that does mean, and we'll hear about the phenotype, using ontologies that other people are using. And again, we really want to interact with the community to understand what are the, and as Alison, another EBI, ex-EBI person, I don't know if she remembers me, there is always new things coming up and things being deprecated, and we need to keep on top of that. So this is the kind of data that we capture, which is quite similar to what you will find in the paper. But the thing is, a lot of this is free text. And we have done quite a lot of work with German colleagues about exactly how much of this instrumentation information needs to be captured. We're working with them on expanding this into further controlled vocabularies so that it is necessary to have all of this information in order to reproduce the studies, which is the key thing. So if you take a look at this, I know it's quite small on the screen, even with my glasses, the level of detail that you have to have for a chromatography, a mass spectrometry experiment is really, really impressive. But this is information that is known. It is really only that some of the machines and the columns will change, but the concept of what you're trying to capture is really important and consistent. I visited a lab last week in Liverpool for the first time in many, many years, and it's remarkable how similar it was. Brought back the nightmares of the heat hoods. Anyway, leaving that behind. The other key thing here is what I would like to highlight is a lot of this information is in the vendor software, and we really want to work as metabolites to pull that together. And we hope to have an industry workshop with Elixir and vendors and experimental scientists, so we can find a much, much easier way of getting the information out of the machines and into metabolites or any other files that the experimentalists want. Because it's painful to write all of this and to think of all of this and to specify all of this when it's all in the machine, and we should be able to get it out much more easier than we do, and definitely not cut and paste. So again, this is some of the level of detail that we need and that we need to control. And that finishes. So giving thanks to the team, which is very small, as you can see, a key aspect in bioinformatics, and some of our funders, past and present. So that's me. Thank you.
Thanks, Claire. I'm just scribbling down my notes and questions. It's a very informative presentation about metabolites. Thanks for the updates. We'll now turn to talk about PRIDE, and Deepti, I think you're on the line, ready to share your screen.
Yeah, so hello everyone. I'm Deepti Kundu and I work as a scientific curator at PRIDE databases at Email ABI here in UK. So today I'm going to give you the brief introduction about the PRIDE databases and that how the workflow and the metadata annotation that what we do here in PRIDE. Can you move to next slide, please? Yeah, so just to give you the very brief introduction about the proteomics that everyone know about it, I'm sure, but just to give you the brief, that proteomics is basically the study of proteome in an organism or tissue or cell, and the approaches used to study the proteome there are different approaches, and the mass spectrometry is one of the main approach used in this field. And since proteomics is very established discipline, the many archive databases have emerged to store the results and the peptide identification results and the raw files for the same. Can you move to next slide, please? So today I'll just give you the brief infrastructure of infrastructure introduction of the PRIDE and the proteome exchange, the metadata annotation of the sets, and then the data reuse of these public proteomic data sets. Next slide, please. So as I said, the PRIDE is the main repository which stores the mass spectrometry based proteomics data set, and currently we have more than 28,000 data sets in PRIDE. And of course, all other proteomics approaches are supported as well. And PRIDE is one of the core Elixir data resources for data deposition and databases. Next slide, please. So, so of course, so we are not the only repository for the proteome to archive the proteomic data set. We have 5 other sister repositories. For example, JPOST, iProx, which are based on Asia, and then Peptide Atlas, Massive, Panorama in USA, and then PRIDE in UK. So PRIDE is the main founding member of the Proteome Exchange, and then we enable the data sharing and which has been generalized in the proteomics field. And the next slide. And just to show you in this graph, the growth of PRIDE since it was established, and it has been growing immensely from over the past years. So we have from like more than almost approximately 500 datasets submitted in every month on an average. Which let alone in 6,000 data sets during 2021. So we constitute the main proteomic data sets in Proteome Exchange, apart from among other 5 sister repositories. Next slide. So you can see the data set which is growing immensely in PRIDE, and we have right now PRIDE constitute approximately 1.6 petabytes of proteome data, and from 2019 to 2020 you can see the growth that it is growing almost 12% in each year, and which has increased like till 500 days. Now it is like approximately 500 data sets on an average. And PRIDE has, and in PRIDE, more than 15,000 research laboratories they submit their data set, and we constitute like 82% of the whole proteome exchange data sets in proteome exchange. Next slide. So this is just to give you the very brief overview that how data set been generated. The proteome data set has been generated by the proteomic scientists, and then the data set then uploaded to PRIDE via PX submission tool, which is our in-house tool, which helps submitters to upload their proteomic data to PRIDE. And then the curators checks the data sets and process the data set to PRIDE, and then data set is at last, at the end, it's submitted to PRIDE, and the accession has been generated to the submitter, which they later include in their manuscripts while publishing their result. Next slide. So this is the basic flow of how the data set being uploaded into PRIDE, and what are the main files needed in PRIDE. So we have the PX submission tool, which is a standalone open source tool, and it also enables submitters to annotate their metadata, but the metadata is captured in very general data. Metadata are captured during then, and then the required files to upload the data in PRIDE are the raw files, which are mandatory files, which is basically the peak list spectra files, and then the result, which is mandatory for the complete submission, and then the peak list file, again mandatory for the complete submission, and then, as I mentioned, the basic metadata, which can be annotated while uploading the data set, and then the SGRF, which I'm going to mention in my next slide. So next slide. So the metadata annotation of public proteomic data set. So we can I go to next slide? Yeah. So till now we were able to capture the basic metadata and then the result and raw files in PRIDE. But next slide, please. But the structured metadata, which links to individual samples to the metadata, was missing, which makes it very hard to annotate for the reusing purposes, and also it would also be it can also be a key for linking the samples in the multi-omics studies. Next slide, please. So we introduced SGRF file formats, and of course we didn't do it from the scratch, because there was a mesh tab file formats already established in this field. So we introduced a new flavor of SGRF which can support the proteomics experimental approaches for specifically for proteomics data sets. Next slide, please. So for now, we either PRIDE curators in PRIDE or collaborators, they can select any public proteomic data set, and then they can annotate it. They can annotate by from the publication, because not many times the whole information is given. So we annotate, we select the data sets, select the samples, and then annotate the data sets from their publication, or if any general information is given in PRIDE, and then we annotate into the SGRF file formats, and then it can be validated in our in-house pipeline, and then the SRF files are validated and are ready to upload into Pride. Next slide, please. So for now, we have incorporated in our PX submission tool, but it's not mandatory right now. It's optional, and it can be tagged as experimental design while when you upload your data set. So but apart from that, we have approximately 450 data sets already annotated for the SGRF file formats in Pride. And yeah, okay, can I go to next slide, please? And then the second part is the data. We also annotate and reprocess the public proteomic data set. And can I go to next slide? So the Pride data set, the public proteomic data set are being reused immensely, and as you can see in this chart, that data set has been downloaded, and the download has been increased over the years as well. Next slide. And you can see that the proteomics repositories, the submitters submit their data to proteomic repositories, and then it being reused by other bioinformatics resources here in IMBIL, EBI, or any other popular bioinformatics resources. And again, these results or these data sets are reused by the community, the scientific research community. Next slide. And here in Pride, we also reprocess the data set, and we disseminate these data sets in different other bioinformatics resources in EBI, for example, UniProt, Expression Atlas, Ensemble, and Magnify. Next slide. So to summarize that, I have explained the Pride archive and the Proteome Exchange, the very basic workflow of PX submission tool, and then how data sets are submitted to Pride, and then how it is available via Pride or the other proteomic repository for the public, and then data being reused, and the reuse has been increased over the years. And the most important, we have also introduced the SGRF file formats, which helps in data reusability, for the data reusability. But it's important because as I mentioned, that we have almost 500 data sets submitted every month, and it's almost impossible for us to curate each and every data set for the SGRF file formats every time. So it's very important if the tool producers, if they incorporate it in their tool, and then they produce the SGRF file formats, which makes it easier for everyone to then use it for later on. And next slide, please. And at the end, I would like to mention our team behind Pride, our team leader, Juan Antonio, who is also present, and who will, of course, help, who will answer the more questions if you have, and our software development team, and then the bioinformatician and the curators team. And at the end, I would like to thank our collaborators and our former team members for the support.
Thanks, Deepti, and sorry everyone for the technical challenges at the start of that presentation. I have a couple of questions, and I'm just going back to my screen to see if there's any questions from the community. But let me start. So, Deepti, so some of the metadata that will be submitted relating to proteomic data set, when we think about human samples, might not be able to be public. So is there a way at Pride, or are there any thoughts within the proteomics community of how metadata could be structured in a fair way that allows the privacy of certain aspects of the metadata, some of it that has to remain under, say, controlled access, remain in that way?
We are going to work on it. Like it has been already in. I think Yuan can answer this more precisely, but we really think about it, and we do have. Yuan, can you answer this more properly?
Can you hear me? Yes. So I mean, Gary, you are aware of this personally. So we have started to design the system to be able to support control access data sets. So we are going to reuse many of the ideas and much of the infrastructure of EGA, but with the particularities that are needed for proteomics data. So the problem is, of course, the technical metadata is completely different. And with respect to the metadata coming from the individuals, then I think we will, at this level, the easiest thing for you to do, the easiest thing for us to do, is to basically do the same that EDA is using. It wouldn't make any sense for us to reinvent the wheel at that level. So yes, there is a high increase in demand for this type of infrastructure, also for proteomics, and also for metabolomics. That I know that Claire.
Yeah.
We can talk about that later in the panel.
And Deepti, to go back to your presentation, this is how, at least I see the possibility of combining informations across a multi-omics data set for the one particular sample, is that the sample is the main reference point and that metadata is linked on that sample. I think Claire alluded to it a little bit in her presentation, but is that the same way that you see it for Pride and for all of the databases that we're talking about here from the EBI?
So yes, so that's definitely the way to do it, I think. So I think Deepti, it was mentioned in the slide, but it was not mentioned by her personally, that we are using the biosamples database at EBI for that, at least for this level of annotation. But something that maybe will come up later in the discussion as well is that we can have the infrastructure to do this, and we can make things as easy as possible. But at least for multi-omics data, I don't see a way to try to link everything together that it needs to be done by a person. It needs to be like a data manager or a equivalent person that is just funded to do that. I think our role is to enable to have the infrastructure to be able for people to do this. But I mean, it's basically impossible, especially since many of these multi-omics studies are done in different labs by different people. There are different types of replicates. The experimental design is different depending on the omics approach. So I think our role here is to enable by having a data formats, having the infrastructure for people to be able to do it. But it is completely unrealistic to expect that just by that it will be enough to have multi-omics fair data, because there is a lot of work that needs to be put on top. That's what I would like to say. Mm-hmm.
Yeah, I think we might come back to that in the discussion, because there's an extra word that we could add in there, which would be federated, which might make it even more complicated. So
yes,
think about that later. Claire, I'd like to ask you a question, because you talked very briefly about a workshop that you're planning with industry. And in my time within EBI and Elixir and now IATRIS, I've spent a lot of time trying to engage industry in these conversations, because both at the data generation and data analysis ends of the pipeline, I think it's really important that we have those conversations. So maybe from your perspective, you could give us your thoughts on how you see the collaboration for metabolomic data working with industry right now, and what the future next steps are to improve the use of FAIR metadata, specifically with industry.
Yeah, so I would echo your caveats about industry, but the thing about the Metabolomics conference is that they're very much there. If you ever go to Metabolomics Society annual conference, half the the space is taken up by the companies. And basically I just went around and invited them all and made it seem utterly attractive, and they've all agreed to attend in principle. And Elixir has agreed in principle to fund it, probably alongside the Metabolomics conference next year. I think what's different now about Metabolomics vendors is, again, I don't know who's on the call, but in the past, they basically had the scientists exactly where they wanted them. Well, now where there's lots of companies that are doing metabolomics services, they have more work to do to show why you would want to do this yourself and why you wouldn't just farm it out. The other thing is, in certain aspects, it is becoming far more a repetitive activity, not just a once-off research project. It's something we've heard about cohort studies, that people will be followed for a very, very long time. So it's very much in the interest of the companies to make their machines be the ones that are chosen, because they will be used and replaced, upgraded on a regular basis. So I'm quite positive at the moment, and it's something that the community, the experimentalists, have come to me a lot of time when they complain about having to put the data into our database. The thing is, a lot of that information is actually stored within the vendor information. They don't always share all of it, so the output that they give, we actually had a discussion about this in Liverpool last week with people who have all the fancy machines, and the output that they get from the machine isn't exactly what they were expecting, because the vendors have thought a lot of it is noise and they don't need to see it, and that's not actually what the users think. So there's all kinds of conversations that we can have there, and I just believe that our team is in a good place to facilitate that, because ideally for us, it's they can sell their machines, the experimentalists can submit to us faster, and we just get the data that we really want and the community wants to reuse. So I think we're good facilitators for this effort, and I fully expect it should go ahead. I know for some of the biggest companies, I have over time got to know them, so I know that they will show up. But for the smaller companies, they needed to touch base with the bioinformaticians back, programmers back at headquarters, because it was more the salespeople that were there. But I really think this will work, and it's about time it did.
Yep. So from the EATRIS perspective, within specifically our biomarkers community, where we have a platform, there's a huge interest in the translational medicine domain to work more closely with vendors for FAIR metadata. So Claire, I'll be keeping a close ear out for when that workshop takes place, and if there's position there for stakeholders, then I think the EATRIS translational medicine community will also be where we have some activity trying to work with industry. We'll be interested in taking part if we can to try and streamline.
No, for sure. Like I said, we're facilitators. We want to bring together all the people on one side who actually use the data, as well as the vendors. Otherwise, we're not going to know what it is that like that conversation I had in Liverpool last week. I wouldn't have known about this problem with the output if I wasn't talking to people who are actually running it in their lab.
Yeah. So I can echo that more from the clinician side nowadays as the users. But yeah, this is a really important conversation that we should facilitate as much as we can. Thanks, Claire. All right, we are a little bit behind schedule, but I think we should still take a break before we move on with the final pair of presentations. So the time right now is 57 minutes past the hour. We'll take a 10-minute break, and we'll come back at 7 minutes past to hear from the final pair of presentations, and then we'll have a panel discussion about the challenges and possible ways forward that we can accelerate the FAIR metadata management. Thanks, everyone. We have 2 presentations left from this workshop, and then we also have time for a panel discussion. In our last pair of presentations, we'll focus on interactive tools to capture and share metadata of multi-omics studies using templates. And the first presentation is from Gilles Baumann to talk to us about the phenotype database as a potential solution. Gilles, can you share your screen, please?
If correct, I'm already, yes, there it is. Thank you very much, Gary, and also Anna for giving me the opportunity to speak today. Indeed, I would like to talk about the phenotype database, and I will show some practical solutions that we have built in, and hopefully it will result in some uptake of these ideas, also in other resources. So setting the scene. So 12 years ago, we started the development of the phenotype database. And at the same time, also the ISATAB solution was developed. And for that reason, the phenotype database was also fully developed on the structure of ISATAB, which was a great standard to stand upon. The reason why we had to develop this database was that, well, in nutritional studies, we have a large background, which means that we need many measurements, so connecting to different omics techniques. But also we have a seasonal effect, and that is why we often have this crossover design where placebo or a treatment is given at other times within the study. And making this type of studies machine readable was one of the goals of this development. Because in nutrition, we are focusing so much on the phenotype and the outcomes, rich metadata storage was crucial. And first, because there was a total lack of good vocabularies and ontologies, but also because of this complex study designs, we put a lot of effort in the user interface. And I will show you today some examples how we worked on this user interface. So first, this is just one of the instances of the phenotype database. I can show later the specific details, but this instance can be used by anyone who likes. They can ask a permission to use, but it's totally open source, so you can also install it on your own server. At this moment, we have 151 studies in the phenotype database, of which less than we hoped is yet public, but that's also because people are very scared to open them nowadays with the GDPR. The whole idea of the phenotype database was to store the study metadata as one component, like also in the idea of alongside the idea of ISATAB, and then connect to different modules, to different databases for omics technologies. So even now we are still running an implementation study within Elixir to show that we can interact with this phenotype data between the phenotype database, Magnify, and metabolites. As I said, I will be very practical today. So I would like to walk you through some of the steps that we have in the wizard to show some of our solutions. So this is the user interface that we're using. And it's a wizard where we have several steps where studies are described. We have some general study information, subject information, design information, samples, essays, and the essays, that's the level where we also connect to the other databases. The important thing is that on every part of this, on every step of this interface, we use templates. So here I'm, for instance, at the step of the study, and I can choose a template, for instance, in this case, intervention observational studies. The one with the green arrow are the ones that are obligatory. If I would have a look at a certain template, just an example on the layer of the design of a treatment, I can select a specific template. And within the interface, I can build up this template by myself. I can add fields to the template. So on this level of the wizard, on the intervention, the name is always there. So whatever template I make, already this field of name is available. And these green fields are the ones that have been added to the template. And the nice thing is that any field that is used in another template on this level is shown here. So I can easily reuse this field, which really stimulates in reuse of the template or of certain fields over templates, which makes the machine readability and interoperability, of course, better. I can also easily create a new field, or at least the template administrator can. And well, we have a lot of types of fields that you can add. So for instance, ontology fields, we have a direct link to PyPortal, but also within the different modules. So the layer where we also store, for instance, clinical chemistry data or dietary data, we can also map our terms to ontologies. As said, I can select different field types. So for instance, text fields like long text, but also drop downs and extendable selection of terms. And those fields are extremely nice because, as I said, we were missing a lot of vocabularies and ontologies within the nutritional field. And a nice thing is by having this type of fields, you can really start building up a vocabulary by just having users adding terms to these fields. And of course, you need to have some control over this, but we have developed in this way some new vocabularies that are really usable. And actually, based on the structure, there's also an ontology has been developed. Then we have numerical fields like decimal number or natural numbers. But as said, also linked to ontologies. We can have a file field, but also a template field, which of course we need to connect to every layer. And then what's very important is that we have a date, which is an actual date in time, but we also have a relative time date. So that means that it's relative to the start of the study. Then I very briefly, in view of time, go over this step of the wizard. The subject step of the wizard is mainly done in an upload format. And the idea is that people normally have the information about the subject in a CSV file or an Excel sheet. So this is normally done by an upload. But what we do here in the wizard is that we connect the titles of the Excel sheet or the CSV file to the fields of the template so that the machine is mapping the terms so that there is an interaction between those terms directly. So what I can do here, I can choose a file to upload. I can indicate on which layer of the file the data is located, also where the column header is, and I can connect it to the template and a specific study. Then I get this overview of the tabular file, and I can easily connect the title in the file to the different template fields that are available in this selected template, and I can also match them, and then the computer will do that for me. As said, we put a lot of effort in the design, especially because of this crossover design. This is an example of how biologists normally draw their design. They have different groups. They have a treatment in time. And in this case, we had different types of treatments. We had placebo and a supplementary mix, but we also had an oral glucose tolerance test and a high-fat postprandial challenge. There are different types of samples connected to the different type of treatments. And that's actually what we have also made machine readable by different layers in the wizard. So this is how it looks like, this study, how it looks like in the interface. So here again, the different groups, here a screening of the individuals. Here you can see the placebo, the oral glucose tolerance test, and the high-fat postprandial challenge. And the machine now understands that this treatment actually is the same as that one, but that it is at a different time actually in this specific group. In this group, the individuals are selected, so they're directly connected to a certain treatment and also to different samples. So from this also the whole sample table is created. If you go one layer deeper, as I already said, the samples are connected to the treatment, This is one layer deeper. So I have now selected one of those treatment groups. Here, the anti-inflammatory dietary mix. And here you can see the different types of samples that have been taken within this treatment, connected to this treatment. As said, I can also connect, I can also indicate which type of assays I've performed within the study. And here you can see also the connection to the different modules. We have developed different modules within the phenotype database, but what is most important for us are the clinical chemistry and the physiology module, because there are really the outcomes measures that are crucial to us. But also, for instance, following BMI in time is now possible because we can collect these data also in time within our system. What is important to say that there are different layers where the data can be opened. So it can be opened on the study design, only showing what type of study you have performed, but also the assays can be made public one by one. There are different ways to output these data. So for instance, you can directly connect to the system via R or Python. There is a search option within the interface, and you can also export the data to a CSV file. And very briefly, so the data is not open, at least not at first. So the person who is including the data in the system is the only one having access to the data. The instance that I just showed you is at TNO, it's located there. But you can also install the code yourself on your own server, and that's open under the Apache license. Because of time, I would like to stop here. Thank you very much, and I'm open to any question. Thank you very much.
Excellent. Thank you, Julian. I'll remind the audience that they are able to submit their questions via the question and answer option within this platform. And after the next presentation, we'll have the final round of questions from the audience on what's been presented. The next presentation we have is from Jory van der Velde on FAIR genomes and Mogenus. Jory, you should be able to share your screen, I think. Perfect, I can see it now.
I think I'm there. All right. Yes, so first of all, thank you very much for having me. It's a wonderful session. So my talk will basically be comprised of two components, this FAIR genomes and Mogenus, but they're very much connected, and in a sense they're actually quite similar to the previous talk. So pay attention, and you will notice. So my name is Jory van der Velde. I'm a postdoc bioinformatics in the group of Maurice Schwertz at the University Medical Center Groningen. And I'll kick off by telling you that in our lab and also in the Netherlands, we do a lot of genome analyses. It's both detecting variation, but also interpreting DNA variation. And that's both for research and clinical use cases. And also, of course, linking the genotype to treatments. And in all of this work, of course, sharing and reusing data is absolutely essential. And in a sense, of course, we're already sharing a lot of things, but it could be a lot more. Because sharing essentially whole genomes and next generation sequencing data is still very difficult to properly share and reuse these types of data. We're dealing with different sequencing platforms, different bioinformatic pipelines, different clinical information, privacy consent issues. So quite often, or actually most of the time, people in the labs are wondering, where is the data? Are they comparable to the data that I have? What questions can I ask the data, and are those questions allowed? That's often, those basic things are not clear. So the FAIR principles here can really help us here. Of course, these principles, you are most likely familiar with them, but if you're not, please look them up. They are very insightful. But sort of where I'm from, we see the FAIR principles as a really handy toolbox to make data better, and not so much as a miracle cure. And this is also probably something to be aware of to educate people. So people are already highly enthusiastic, but other people might not be so, might even be skeptical or even a bit hostile to these principles. Maybe they had some bad experiences, they expected too much, and then things didn't turn out as well. So we've had these experiences, and I would just like to make you aware of the fact that often the challenge is not so much technical, but rather social. So just as a small take-home message. But onto the actual project. So the Fair Genomes project, we involved 66 people from the Netherlands, from 14 different institutes that all deal with NGS data in one way or another. And then we asked the people at the actual workplace, so the people doing the work, like what do you need to know about next generation sequencing data in order to reuse it? And from all of those answers that we got, we organized workshops, did interviews, meetings, and we then built this metadata schema of the most important properties that we should capture about these data. And of course, this was iterated many times. And based on this, we created pilot systems and had some test drives to see what worked and what didn't. And of course, the final step 5 would be to apply these metadata internationally to achieve a higher degree of fairness and be able to share and reuse data better. So the actual building of the schema, this is what we did. We defined in the end for this type of data, for this domain, 9 modules. So they will sound very familiar to you, things like personal, material, sequencing, clinical data. And each of those modules has elements. So essentially, modules are like database tables in a sense, and elements are like attributes or columns. Like, so for example, for clinical, you could have a medication or age of onset or consanguinity. And then preferably each of those elements that are then also given coded value lists. So no free text, of course, but properly coded values. So for phenotypes, an obvious choice is human phenotype ontology. But absolutely everything in this schema was coded with ontologies. So the one advantage is that you're then actually very precise about what you mean, because you can look up a very detailed definition. And the other one is, of course, that it enables computers to talk to each other in an unambiguous way. And we also added so-called null flavors from HL7 FAIR, and that's mainly to make it much more powerful to explain why certain values are missing. So instead of just leaving things empty, when you are filling a database, you can then explain why a certain value was not there. So that also gives you sort of a semantic richness, even if the data is not available. And all of this is, of course, you can look it up. And of course, the schema is nice, but you need to do something with it. So, get practical. So we wrote a little generator that was able to sort of translate this schema, which was defined in something called YAML, into a variety of electronic data capture software packages. So things like RedCap or Castor, OpenClinica, and also Molgenis, and also documentation formats. RDF that we were also able to submit to places like Fair Sharing and Bioportal. So basically, all of these things are called, are sort of artifacts that are derived from the schema, and so you don't have to use one system. You can use whatever you like, but you will still be interoperable because you're using the same definitions. That was sort of our goal. And well, if you want to know more about this particular project, then I would highly recommend to of course read the paper or visit the website. I have a tiny URL at the end of the presentation, so I'm not sure if you're able to share the slides, but then you can just use this tiny URL to look up the talk and get all this information. But for now, I would like to continue a bit about this Molgenis. So this is one of those electronic data capture systems. So this is being developed in our department in Groningen. And it's not something new. It's actually been used already for over 15 years. And currently we have about 100 servers running for all kinds of different customers and applications. The software itself is free and open source. So we mainly provide, let's say, the software as a service component. And currently the 5th generation of this platform, so we keep evolving and adapting, is being built. It's codenamed EMX2. And the rest of the talk will be about how this EMX2 will help to make data more fair. So obviously, it's very easy to put data in. So there are some standard formats for what you could call metadata, which is basically the data structure. So for example, if you have a table called analyses, then you want to define all of the strings and integers and data types, just as you saw in Yildau's talk. And then, of course, there's the actual data itself. So the rows of your tables are then entered also in very simple formats. So nothing special here, but of course it's there. And once your data is in, you can then start to filter and query and sort and whatnot, as you would expect from a relational database system. One cool thing, though, is that Multigenus always has been a very flexible. So it's always possible to change the underlying data structure. So you can add columns or change data types on the fly. And this is basically because you're often at the start of a project never sure how your data will exactly look like. So you need a bit of flexibility to adapt to new metadata or new standards when that need arises. And of course, we offer many APIs to get data in and out of the system. So stuff you would expect, like CSVs or Excels or JSON-LD, Python. But for now, I would like to focus on these two, something called Beacon v2 and something called Fair Data Point. So I have no clue if the audience is familiar with these things, so maybe a very quick refresher. Fair Data Point, I would define that as it's essentially an interconnected set of RDF documents and usually served by an API, but not necessarily. And it basically offers you some machine-readable, actionable metadata about what is present in the actual database to basically regulate the discovery and access to these data. So it's quite lightweight and quite elegant. And this Beacon, on the other hand, has two components. So one is it's basically a JSON-based API framework for discovery of data, but it's also a domain model describing DNA variation and samples, clinical data, and so on. And there's many options to deploy this Beacon and then run federated queries on what is called the Beacon network. So these are two very popular options to basically make your data known. And so what we've done is when you create a database, and here again, there's the template aspect coming back, except this time it's basically on a whole database level. So there's a template called Fair Data Hub, and you can load this template. Instead of an empty database, you now have a number of tables already in there from the start. So there's a number of these you can see on the left. So analyses, biosamples, basically those are Beacon related. And there's also two called, starting with NTB, those are Fair Data Point related. And they're all using these sort of shared standardized ontologies. And this gives you the structure that works with the APIs that are also present in these in this platform. So what you do, for example, I want to create a new Fair Data Point catalog entry. Then you add just a row in a table as you would normally. You can either do this by a form or also by a CSV file, etc. And once and you can use the standardized ontologies. So here's an example for diseases. So that's also pre-packaged with the system. You don't have to worry about how to code the diseases or disease severity or whatnot. So what you get, you instantiate, basically you run this EMX2, you use this database template, and at that point, you already have a working Fair Data Point, a working Beacon, a working GraphQL endpoint, which is basically a very clever API gateway. And of course, you can then load in all kinds of standards like fair genomes or semantic common data elements or ISOTAB, whatever you want. And let me summarize it for you. What you need to do as a verification guide is basically to start a server, run this template, and then add your own data instead of what is there. You probably want to delete some of the example data after you figured out how it works. Set some permissions for discoverability of your data. And then once that's set up, you can just register your FAIR endpoints. So there's places like the Beacon network registry or the Fair Data Point home. And that's probably a good way to make your data known to the world and also have it a part of using these standardized ontologies behind it. So that was what I had. And of course, we're not doing this alone. So we're collaborating a lot with projects that also work in this space and touching on each other. So that's why it's really excited to be here and give this talk, because I've heard so many of these things fly by. And of course, together we should find out how to share and reuse these omics data in both research and healthcare. I think that's very important. So thank you very much for listening. I'd like to acknowledge my colleagues and partners. And if you want, you can you can find this talk at the URL I put in the corner of the screen. And yeah, I'm looking forward to your questions. Thank you very much.
Very good. Thank you, Yuri, for this presentation on Mulgenus and FAIR genomes. You can see there's some clapping hands going up the screen right now, so I'm not the only one that appreciates it. Very good. Can I ask a question? So for the Beacon V2 network, what kind of queries are you commonly seeing for the Mulgenus installations? What are the most common things that people are looking for? If you have a feel for that, it'd be really quite interesting to know, because that gives you a real deep view into the user perspective of what people want to actually find, samples or genetic variants or whatever else. What are the common queries that you see there?
So right now, I would say it's phenotype-based searches. So people want to find other patients with similar phenotypes, for example, in different European reference networks. And the other is variants. So people wanting to find if a particular variant is present in another center, and even preferably what their clinical opinion was of this variant. So I guess those are the two main ones.
Perfect. I thought you might have said something about the ERNs. So how do you see the uptake at the ERN level for, say, Mulgenus with Beacon V2, and the complexity that one tries to bring for quite serious technology into say the ERNs? Has there been a huge uptake of this, and maybe what do you see as the biggest bottleneck there?
Yes, they're very eager to use these technologies and share their data, but for ERNs, they're also very careful about not sharing too much. I think that's where Beacon can be of big help, because you can easily restrict the access to the Boolean level, where you say only allow discovery of the fact that something has been measured and not the actual patient data itself. So that will help. And within that, it's indeed all of the clinical details mostly that they're very interested in sharing. Thanks.
And then Gilles, right at the very start of Yuri's presentation, he mentioned something about the connectivity between the phenotype database and what he was about to present. So could you comment on the connectivity between the resources that we've heard in the last two presentations?
Well, from my point of view, I think the flexibility on the back end of Mulgenus is the very strong part of Mulgenus. And I think the front end is the strong side of the phenotype database. So that's also a way that we are discussing the interaction, that we really think that the interface and the way that we really interact with biologists in general can help filling the very nice, flexible database structure of Mulgenus. So I think that would really join the two strengths together.
Excellent. Jory, in the question and answer panel, there's a question that's come in about how does the FAIR genomes data model align or interoperate with the MIXS standards? If you can see this.
I can't. That's in the live Q&A.
Yeah, it's in the live Q&A panel.
Ooh.
If you don't have a comment on this now, I'll leave you to provide a text.
Yes. No, so we've had a look. To be honest, it's a standard that we were not aware of until quite late in the project, I have to admit. But we've also had interactions with the Genomic Standards Consortium, and we've taken some of their advice to heart. So, for instance, they mentioned that it might be a very good idea to make it less human specific, because now we know it's all centered around humans, but there's actually no real reason why that should be. You can easily extend some of the code listings to fit other organisms, and that might make it much more broadly applicable and allow finding data in a much broader space without too much changes. So that's something we plan to do. Yeah.
Very good, thank you. Thank you both, Gilles and Jory, for these excellent presentations. So now I'd like to move on to the next and final part of today's workshop. So we will have a panel discussion. So all of the speakers that we've heard from today, there'll be a discussion across all of us about some of the most commonly seen challenges in FAIR metadata. So Paula, if you could put all of the speakers into the window. There we are. We're all there. Hi everyone. And what I'll also do for this part of the session is try and bring audience participation in. So I will share my screen, which will show you a Mentimeter page. So for those of you that aren't familiar, Mentimeter is an online platform where the community can provide text answers to a question that's on screen. And as the panel discuss a particular question, we're able to take answers from the community into the Mentimeter platform. So let's try that. So if I share my screen and I show this Menti question and share that in a presentation mode, hopefully everyone can see the question. First of all, at the very top of this page, there is the directions of how to provide your answer. So if you go to menti.com and type in the code 22773388, that will bring you into this live platform where the audience can provide their answers to this particular question, which is, what do you think are the main bottlenecks and challenges in the fields of standardized and verified multi-omics metadata? And as you start to type your answers there, it should pop up on this screen. But for verbal answers, I'll turn to the panel and ask anyone that would like to step in and provide a first thought on the question here about the main bottlenecks that are seen in the field of standardized and verified multi-omics metadata. Who would like to kick us off? Showdown, I see a hand.
Yeah, at least I can kick off with some things that I think are very important there. So I've seen that for rich metadata, still, although ontologies are developing rapidly, we still lack quite some ontologies. So that would be one bottleneck. I think the LC aspect and the psychological aspects are very relevant still for getting well verified data. And what I mean there is that, especially for human studies, there is still the people are scared of the GDPR. They do not understand how and which circumstances they can open their data, or at least how they can be made reusable. But also the psychological aspect that I'm referring to is that the people feel very scared to open their data because they are afraid that they will lose their publications because of that. They think they can publish themselves more about those data, so they prefer still to keep them close. So even if the omics data is there, a lot of the outcome or the other rich metadata are still lacking in the repositories. So you cannot really reuse this data. Yeah, I think that at least are the first points that came up in my mind.
Thanks, Yodel. Absolutely, the social aspect of sharing data is still a challenge that we must overcome in all fields, including metadata verification, and trying to provide trust in the systems that we offer. Juan Antonio, I see a hand.
I mean, so for me, there are two aspects here. One is, I mentioned it before, is that the scientists need to be incentivized somehow to provide and annotate their samples properly, and especially multi-omics studies, and collaborate. And it needs to be seen like a natural part of the project, all these efforts that need to be done in this direction. And at the moment, I think that what is valued is the publication, as we all know, and all this is secondary in many cases. Or the other option, and maybe a complementary, is again, there need to be resources for it. So that means for these projects to have a person that needs to be paid to do this job, because otherwise, I mean, we know that it's pretty complex and this will not change. It was mentioned before that many of these fields are very dynamic. Even if we have a standard, things evolve. The tools will not be in sync. So I think that a big part of it is also to recognize that resource is needed to do this properly. So at least from my perspective, and I don't see that properly recognized. So that's the other part of my answer.
Thanks, Juan Antonio. Yeah, these are huge. These are huge problems, not only in terms of the incentives, but I really like your answer on the availability of resources and tools that allow scientists to easily verify their data. This is and the sustainability of these resources as well, which I know is a subject close to many hearts on the call here. Anyone else from the panel would like to give their thoughts on this question about the main bottlenecks? Claire?
So I would echo Gilda's comments and Juan Antonio's, but I think the other issue there is, and it's not so much related to the papers, but it's more people don't yet feel confident that they're interpreting the data properly. And so often they're holding it back, thinking that something else will come up that would help them understand it. I still, at least in the field of metabolomics, there's still a kind, not a shyness, but a reticence about sharing data because it's quite easy to get it wrong. So one of the things I recently got funding from the BBSRC and the NSF was for tools to be run over existing data to see if the predictions of the compounds is correct or up to date. And I mentioned in my presentation the fact that the compound libraries are often internalized. So I was thinking about this in a really positive way, that data that was a snapshot at one particular time could be reassessed over time as the libraries increased. What I didn't realize was that my community got panicked because to them it would highlight the fact that they had potentially got it wrong. And I've had to do a piece of I guess not PR work, but reassurance that it's going to be flagged with evidence attribution saying this is a program. You know, they did their best. The programs are doing their best. It has to be evaluated on an ongoing basis. And I think that's another limitation. People only share what they feel comfortable to share.
Yeah, that's a nice point. And I've seen similar in the communities that I work with, both from Beatrice and Elixir. And maybe the way that I try and spin it around is always in the collaborative sense. So, you know, we present results, and we don't present results to be criticized or to be harshly dealt with, but it's about furthering the conversation. One's interpretation, then compared with another one's in the form of collaboration, can lead to new analyses and new results. But it is absolutely something that I've seen as a hesitancy in terms of people wanting to share their data more publicly. Thanks. Any other thoughts on the main bottlenecks that we see in this field? Philippe?
Yes, what I found interesting was the recent announcement by NIH to somehow mandate the release of the data. So I think this is also showing a kind of, maybe this is more the stick approach, but having a stronger voice from the funding agencies to have these policies in place, but also not only to do this, but also to provide the necessary support to develop the standards and the tool. So that would point towards coordination, which would avoid the fragmentation of some of the, we can see a lot of will reinventing, or at least it's just simply because it's been highlighted in fair sharing in a way where you can see the communities that are in a way developing similar things, but not talking to each other. And this is where the support to standardization would, in a way, probably do a lot of good, as well as having possibly a more forceful mandate to recommend standards. I think the fact that there is a bit of a vacuum sometimes causes, loopholes, and people can use this. That's maybe a bit drastic, but that would be the position.
That's very good. So I do spend some time, and I'm sure a lot of us spend some time with the policymakers to try and drive towards policies that would enable verification of metadata, verification of all data, if you like, or as much as possible. Okay, I'm a bit conscious of the time, so I'm looking at the screen here and some of the answers that have come from the community, and there's some overlap from what we've heard in terms of incentives for scientists, lack of coordinated development, short-term initiatives. One of the ones that I also see on the screen here is the awareness of tools and resources available to researchers. And absolutely, I see that in a number of different places. So it's great to hear from, for example, Alison today about fair sharing, about registries that are there where people can go and put in their use case of interest and find resources that are connected via policies, via standards, via different tools to try and help that. I now want to move to maybe the more challenging question, which is what are the necessary steps to overcome these key bottlenecks and challenges in the fields of multi-omics, metadata standardization, and FAIRification? This is us looking at maybe the solutions that we see as possible to try and overcome these key bottlenecks. Who would like to kick us off with an answer on this one? Alison?
Thanks. Yes, I appreciate that. I was at the Research Software Engineer Conference last week. It was very interesting because they were talking about the same thing. And one of the things they mentioned was how important it was to, from their point of mind, for inserting a data steward, community manager, a fair data champion, perhaps you might call it, in your project from the start. So you're thinking about FAIR from the start and looking at your funders or institutional policy, check with your library or your project's data steward, to ensure the work you do is FAIR ready from the start. And some of this, maybe another angle at this is looking at the resources you want to use and check and see if they have the criteria that you need are present in those resources. A lot of work recently within the RDA about data repository attributes and things, so that those can sort of align and you can search amongst those attributes for the resources you need.
Yeah, absolutely, Alison. I agree with all of this. Anyone else have an idea on solutions that we could work towards to overcome the bottlenecks?
I really like the data steward point, but I would like to, from experience, I would like to reiterate that this person has some kind of power, because quite often what I see in big consortiums is that the idea is there in principle, but that these people, I've seen it in the States and NIH-funded grants, they spend their whole time running after the scientists trying to get them to do it in the right way. It needs to be written into the grant as harsh as possible that this is a deliverable, not a wishy-washy thing, because I have seen serious frustration. Those staff are trying to do their very, very best, but without the material, they can't deliver. And they're very aware of it as well.
So can I add something to that?
Of course, yes.
So, thanks. So yeah. So indeed, there's this you need incentives to do people verification, but it often feels like sort of an afterthought. Like, oh yeah, we also need to do data verification, and it will take us an extra person, and we don't have that person. So indeed, I would sort of urge for a shift towards tooling that makes data fair at the source. So basically, when you're collecting the data is already encoded, because there's no other way to do it, instead of do it after you've collected and done the project. That's nobody likes to do that. And maybe with regard to the other point, so the fragmented resources and tools and standards, indeed, that's a very important issue, because people are completely confused on what to do. But maybe the natural solution will sort of occur on its own when really big projects and consortia start to adopt specific standards and tools. I think we're maybe seeing that, for example, with Beacon, where people are picking this up because it's been pushed by, for example, Global Alliance, and people start using it and deploying these beacons. And then such a solution will take hold. So yeah, and then let evolution take its course.
Yeah, thanks both Claire and Yuri. And just as I think about this session that we're having today and the next steps, I'd like to try and write this up as a short paper. So if we can come back to you for best examples of where you've seen this in practice, that'd be something really nice that we can share with the community, I think. Any other thoughts or comments from the panelists on other solutions that we can offer here? So I see Anna and I see Gilda, so I'll go to Anna first.
Yes, yeah, also I agree with what was said before, but I'm also thinking that the different research fields, the subject areas can be so specific that it really requires the expert knowledge to know what the data means to make it fair. So a data steward that is responsible for a wide range of subjects might not have that expert knowledge. So I think it is key to enable the researchers themselves to make their data fair. And in that regard, I really like the tools that were presented by Yuri and Yilda that, yeah, as a step to have a more user-friendly interfaces for researchers to also apply these schemas that are being.
Absolutely. That reaches into the kind of sustainability of the whole framework as well. To actually provide tools for the researchers to do this at source helps. Gilda?
Yeah, I wanted to extend to the comments before. I think what is also necessary besides this data manager, that indeed jointly, like Anna indicated, with the other scientists in the project is making those data fair. It is crucial that already, maybe even in the consortium agreement, it's very clear when the data will be made reusable to the outside world. Because the problem is that the most often it will only be opened after publications. And that's very often already after the finalization of the project. So who is then going to decide that the data is open? I've seen many studies where the idea was really to open it up. But then the question was, did we publish all the articles we planned? Can we now open it up? And then, of course, there are all these open articles that have not yet been published, so people keep it still closed. So I think a real clear agreement early on is crucial there. And then I also would like to refer to what Juan was saying before, that there should be a return on all the investment. If people have put so much effort in opening the data, what will they get back from it? So I think having an agreement, what will happen if your data is used by others, that's, I think, key also in the point that I made before in the social part of the interaction. I think that's really necessary.
Yeah, absolutely. Very good. Any other thoughts from the panelists on solutions? Philippe?
Yes, I think great points by the members of the panel. Two thoughts on the initiative that I saw in the US. One is the Open Science Foundation and the pre-registration initiative that, in a way, that was to make sure that people would describe their experiment even before it started, and also specify the kind of analysis they would run. So focusing on the study design and the statistical treatment of the data. And this is almost allows you to almost run a kind of dry run of your experiment and see what you could produce. So it would be an interesting aspect if we could combine these things with the point that Claire was doing, like much mandating clearer ways of guidelines for managing the data. That would also facilitate the deposition of the data, just to address Gilda's point, to have this registry of what is intended to be done. It could be kept, obviously, under wraps with access control, but at least we know ahead of time that it could be managed that way. It's not always possible, of course. I know there are studies that do not lend itself to this kind of treatment, but I think this is showing a nice indication of what could be done, and maybe this is almost a kind of digital twin of the study that could be made available. Again, research to be done.
Very good. Juan Antonio? Juan?
Yeah, I just wanted to add the idea that I think it would make much easier also live for verification, that some of the most popular analysis tools in the different fields support the metadata standards, so that the scientists, when they are using these tools, it's basically the same idea that was mentioned before, is that the most difficult part is already done at that point. Because what is very difficult is once the data have been analyzed, the paper has been written, come back to notebooks and and similar things to figure it out what each sample was. So I think that's one particular problem, is that many, at least in proteomics, never the analysis tool has really paid too much attention to the sample metadata. I think this is starting to change. It's a slight change, but if if most of the annotation will need to be done at that point, and then the tools could export the metadata standards, then most of the work could be done at that point. So that's I think that software developers and tools also are an important part of all this ecosystem.
Fantastic. Yeah, I agree. So we have just over 2 minutes left of this session, and I want to turn to each of the speakers in turn and see if they can give one message for the community that we have on the line here about the key takeaway that they want to present, they want to think about in terms of FAIR metadata. So I'd like to hear from about 15 seconds, please, from each of the speakers. And if I go through you in turn, then that'll give you a chance to give your 15-second summary. So Anna, what do you think should be the key takeaway of this session for the people online? You're on mute.
Findability of the standards. Oh, no, I'm okay. Findability of the standards, then user friendliness of the tools, and what I've gathered from the presentations, the maintenance of the tools on the development.
Yeah, thank you. I'm just going to go through you in order of my screen. So Yori, you're next. What would your summary be, or your main takeaway?
I would urge everyone dare to invest in FAIR at the source data instead of fixing it later. And if the tooling to do that is lacking, then get in touch with the FAIR community and also invest in the proper tooling that is needed to either create new tooling or find the right existing tooling to make FAIR the default way of working.
Very good. Gilda, I turn to you next.
Yeah, I would like, in addition to what has been said, I think we need all of you. So all the listeners today, we need all the people in the scientific world to make this solution working.
Thank you. Veronica?
Yes, I think having data FAIR in every project is very much collaborative effort. So the communication and keeping people informed of what's out there and develop more standards for them.
Thanks, Veronica. Alison, you have a final takeaway?
Absolutely. So I wanted, sorry, the dawn from the time extended really. I wanted to repeat what I saw in a Twitter thing from about 2011. It's not my music, is it? Oh my goodness, it is. Something's playing in the background just as I'm about to talk. Let me see if I can find it. Apologies. At first I thought, because it happened just as the time went on to 0. Ugh. So I wanted to say, in a tweet I saw in 2011 from a guy called Jason Scott, metadata is a love note to the future. So I want everyone to just write their love notes really well from the very start, as everyone said, and not just the metadata fields, but remember those relationships and how everything works together and connects.
Thank you. And the music was very poignant. Philippe, would you like to give a comment?
Yes. Well, again, I think it's FAIR by design, but it's moving things upstream as much as you can, in avoiding self-inflicted pain, and try to exploit as much as you can the study design to drive the creation and the compliance. I think that would be a really, really nice things to achieve.
Absolutely. Juan Antonio, final comment from you?
That change of mentality is needed in many people for this to happen. We can be preaching here as much as we want. If the scientists don't see this as something useful to do, it will keep failing.
That's a really nice one. Thank you. Deepti, you have a final comment for the community? I think you're on mute, Deepti.
Yeah. No, as a curator myself, I do curate or annotate this, and I know the pain, how difficult it is to do that. So as Alison mentioned, we all want the love note from all the scientists when they upload their data set. So yeah, that's it from myself.
Thank you. And Claire, a final comment from you?
Again, it's the same thing. We are an ecosystem of people. So I have been in curating bioinformatics for many years. And please don't ever hesitate to reach out by email or Twitter or wherever to the communities that you want or the resources that you want to know something. Don't just sit there on the other side of the computer going, this is too hard. Just get in touch with us. We can't make it better if you don't tell us what's annoying you. And often there's an easy solution. So on behalf of Metabolites and the curators in my team, just ask us. And the software engineers always respond far better if they get a requirement from our user community than from their curators who sit in the same office as them. So remember that. It's never a lack of time, a waste of time to email us.
That's a really nice way to finish, with the community spirit in mind. Thanks so much, Claire. Thanks so much, everyone, for all the speakers that attended the session today. I think this has been a really fantastic workshop. I've tried to note down as many comments as I can, and as I've alluded to a couple of times, it'd be good if we try and write this session up in some sort of brief communication so that we can take what we talked about here and share it not only with the people online, but with the wider community. So thanks, everyone. I hope everyone enjoys the rest of the ECCB conference. This has been a great kickoff early in the meeting, and I wish you all a good evening. Thanks for joining. Bye-bye.
Thank you.
Thank you all. Bye.
Thank you. Bye-bye.